So let's hit it. Now, I was a little concerned when I asked the question I asked earlier and got all of the blankness. Yeah, they not. Can you see over in the corner? Yeah, you're going to fall asleep. That's the problem. That's the problem right there. It's all right. I'll talk aloud. Huh? Yeah. Yep. We have place for 2, 4, 5 more people, I guess. All right. Before we go on to the special senses, we evidently need a little bit of a review. So when I talk about a nerve impulse, I asked the question before. You've had a few moments now to gather your thoughts. Let's review what a nerve impulse is. Some sort of stimulus is going to come in contact with receptors on the dendrites of a neuron. Is this ringing a bell? What's going to happen next? Nobody's traveling anywhere until this happens. That receptor on the surface of the dendrite is going to be affected somehow. What's going to happen? No, neurotransmitter is going to cause this to happen. That might be one of the stimuli. That's a neurotransmitter. We're past that. Because when we talk about this chapter, it's not necessarily a neurotransmitter. It could be what I'm doing right now. What am I doing right now? Speaking, yes? That could be the stimulus. Mechanoreceptors. Where are we going to talk about those when we talk about special senses? Where are they located? Nobody even looked at the chapter. Holy moly, you had way too much fun. In the ear. In the inner ear, when we talk about our sense of hearing and our balance. Those are mechanoreceptors. Some sort of mechanical stimulus will cause this to happen. You still haven't answered my question. What happens to that little receptor on the surface of the dendrite when it comes in contact with its stimulus? Not yet. Because what has to happen to the receptor? It changes. It opens up. Here we go. We're getting there. And what comes in? Sodium! Yes! So sodium rushes in. It causes what to happen? Depolarization due to a change in what? Charge. The charge inside the cell, the membrane potential associated with a resting neuron is going to change. Inside will become more what? Because sodium's rushing in. More positive. And that change has to reach a certain place on the neuron in order for it to change the whole neuron. What's that certain place on the neuron called? Threshold is the point, but what's the physical place on the neuron? The axon hillock. Very good. Has to reach threshold. So whatever threshold is the change in charge that's then going to cause all the rest of the little receptors to fly open. Remember we had two different types of receptors? We had a chemically gated receptor and we had a voltage-gated receptor. So we have to reach threshold, that certain point at the axon hillock, to kick open all of the voltage-gated receptors we're going to find on what part of the neuron? That big long piece that hangs off the end. The axon. Yes? Then depolarization will take place. Once that change reaches the end of the neuron, what's going to happen next? A stuff, yes. It's going to cause the neuron to do what? Release the A stuff. What's the A stuff? No. Acetylcholine, which is one type of receptor. What's acetylcholine? That acetylcholine is going to cause the change to happen in the next neuron down the line. Is that a hint? What's acetylcholine? Nothing? I got nothing? It's a neurotransmitter. Very good. Whoa.
I wanted to say it, but I was like,
Wow. See, you have to get used to speaking in a smaller room. See, when you speak in the big lecture hall, it's like, okay, if I don't say it right, maybe she won't figure out it was me. You just go ahead and say it. So, that's what we have to remember. So when we talk about the special senses, we're talking about a group of organs in the body that house special receptors for special stimulus. And typically we're talking about, something that happens up here in the head, our senses. And that's why we call it special senses. So the first of the special senses houses receptors that are going to be stimulated by what? What's the stimulus for these receptors? Who? Light. What kind of receptors are they? They're photoreceptors. So when we talk about the eye and vision, we're going to talk about the parts that protect the photoreceptors that are responsible for sending information to the brain so that you can do what? See. Because where do I see? In your brain. The eyes are going to send the information, but the brain is going to process that information. So if any of these pathways are blocked, I might not be able to visualize properly. So in order to visualize something properly, I have to get the stimulus just at the right point so that it can sufficiently stimulate the receptors for vision. And when we talked about the receptors for vision, there's two different types. We have ones called rods and we have ones called cones. And they hang out in an area of the eye that we're going to talk about in a minute called the retina. So all of this other stuff we call accessory structures of the eye. These are things that are going to help get the light where it needs to go, and they're going to help protect those receptors so that they stay intact, so that they can send messages to the brain for vision. So we start off talking about accessory structures of the eye. And the first one they talk about are eyebrows. Believe it or not, those are important. Why do you think you need eyebrows? Why is it our eyes are kind of pushed a little bit back in our skull? Yeah. You ever go into like an older house? This is probably where you can see an example of it the most. When you go into an older house and there's sun beaming through the window and all of this stuff is floating in the air. That's where you can actually see it the best. But that happens all the time in the environment that you're in. So there's constantly things falling down to the ground in your environment. So that's why your eye is kind of sunken in a little bit, and your eyebrows are there to help protect the eye. The outer portion of your eye is exposed to the environment. So some of these other accessory structures are going to help keep that outer eye moist and protected from all that particles, all those particles that we find in our environment. So next in line is the eyelids. You have two of them. They close and open. And we have a bunch of different things there that are going to help protect your eye, that outer exposed portion of the eye. We have glands in there that are going to help secrete liquid, glands that are going to help secrete oily substances. so that we can keep opening and closing the eye. So I'm not going to get crazy about muscles in this chapter, but I do want you to know some of the different parts of the eyelids. We have, in this diagram, we can see glands like tarsal glands. We also, another accessory structure of the eye is conjunctiva, membranes, serous membranes that produce what? What do serous membranes do for a living? That was from last semester too. Serous fluid, right? When we talk this semester about serous membranes, we're going to talk about a whole bunch of different ones. When we talk about the cardiovascular system, we're going to talk about the pericardium. The pericardium is a series of serous membranes that makes what? Pericardial what? Fluid. So that's what serous membranes do for a living. They make fluid. We call them different things depending on where we're talking about. If we're talking about the lungs, we're talking about pleural membranes, and we're talking about pleural fluid. If we're talking about the abdominal pelvic cavity, we're talking about different serous membranes like the mesentery and the mesocolon, and we're talking about abdominal ascites or abdominal pelvic fluid. In the eye, the conjunctiva produces fluid to help moisten that part that is exposed to the outer surface. Correct. What's conjunctivitis? Inflammation of the conjunctiva. Now that inflammation can then sometimes mess up some of these other glands that produce almost like a oily substance. That oily substance helps the eye to open and close, and when it closes for the night and opens back up, it helps to lubricate that surface. It also acts as an antibacterial agent. But when I have conjunctivitis, inflammation, or because usually of some sort of either viral or bacterial contamination, I get inflammation of the conjunctiva. Anybody ever have conjunctivitis? You wake up in the morning, you can't open your eyes because they're all crusted shut, because those other glands that normally would allow that nice lubrication to take place can't do what they do for a living because of that inflammation. So the tarsal glands we see discussed here as well, and the tarsal glands will help with that open and close action. Eyelashes, the heck are those for? No, girls, they're not for putting mascara on. I could never understand this. Okay, the eyelashes are there to help protect particles from going into your eye. But every time I try to put mascara on, what do you think I do? I poke myself in the eye. It's like, this cannot be good. This cannot be right. Right? So that's what eyelashes are for. Not for putting mascara on. Huh? Yeah, no. I do it like maybe twice a year, and usually, I poke myself in the eye. Yeah. And the other thing is, there's bacteria that hang out on those eyelashes. So for me, who uses mascara maybe once or twice a year, tops, guess what? I have to buy a new mascara every time I use it. Because when you take the bacteria off of the eyelashes and then stick them back in the tube, guess what they do? They have more bacteria. So if you use mascara, ladies, you should make sure you replace it frequently because it can cause you to get infections. Don't share things like mascara, because chances are you will give somebody else conjunctivitis and they won't be happy with you. So that's what lashes are actually for. And then they talk about the tarsal glands, again associated with the eyelids. They are going to help with the opening and closing of the eye by producing substances that are going to help keep you happy. Conjunctiva membranes associated with the eye, producing fluids, keeping things moist for the part of your eye that's open to the outer world. The other big producer of fluid that's going to help keep your eye, the part that's exposed to the outside world, most of your eyeballs are inside your head. Do you remember the bones that make up the orbit of the eye? Lacrimal, I'm so glad you said that first. Why do you think the lacrimal gland is what I was so excited about you saying first? Lacrimal bone, excuse me. Yeah, well, the lacrimal glands, the bone is named for the glands that sit right behind them. The lacrimal glands are going to help, well, at least the outspurting of lacrimal glands, or the drainage point of lacrimal glands goes through the lacrimal bone. But you have glands that sit above in your eye that produce fluid that sort of squirt down and shower your eye to help keep it protected as well. Again, these fluids, when we talked sweat, for example, in the integumentary system, these fluids contain substances that are going to help act as antibacterial agents. So when we talk about lacrimal gland secretions, what are they? What do we call them? Tears. And inside those secretions, we're going to find things called lysozymes. And lysozymes are acting as antibacterial agents to help protect the eye that's exposed to the outside world. So those secretions then wash across the eye and drain into what? What's this? This is the reason why if you go on your first date, don't go to a sad movie. Extremely unattractive. Do you know where I'm going with this? Because when you overproduce tears, and you do that sometimes, the nervous system causes you to do that when you're sad, guess where they're going to drain and not quite nicely go down the back of your throat indiscreetly.
So that's why when you cry a lot, you get something else.
Yes. Or you get, yeah, not attractive. No. So go to a comedy, first date comedy.
Yeah, but if you make her laugh, then you can cry.
Oh, that's true. That you cry too. But it's a little bit more attractive, I think, when you have things coming out of your nose and you're actually laughing about it. So the lacrimal canaliculi is a drainage for those tears, which normally happen at a very slow rate. And what do I end up normally doing with them? I swallow them all day. Yeah, I know it's gross, but yes, that's what you do. Unless you overproduce them, then you have to blow your nose. But that's where the drainage point is for the lacrimal glands. What kind of gland is a lacrimal gland? Since we're going into this in the next chapter, I might as well hit you up with it now. It produces something way up here, and then it shoots it out these little tubes somewhere else. What's that called? It's an exocrine gland. Yes? The next thing the book talks about is those extrinsic eye muscles. Again, I'm not going to get crazy about those. If you look on page 547, you'll see a bunch of different muscles that move the eyes in different directions. We have binocular vision. What's that mean? Yeah, we use two eyes to gather information for sight. And we move them in unison. At least we're supposed to move them in unison anyway. And these muscles are going to help us do that. Again, we're moving them so that we can position ourselves just right so that light can come in at just the right angle and hit the receptors at just the right intensity so that we can process what we're seeing in the brain, vision. So I'm not going to get crazy about them, but we have a bunch of them because we're able to move our eyes in a whole bunch of different directions. There's some on the top, there's some on the sides, and we're good with that. Now, we'll have a peek on the inside. Who has lab today? You'll be so excited. Guess what you're doing? Dissecting an eyeball, yes. One of my favorite labs. There is a link, well, on your activity sheets. Has everybody seen your activity sheets? Has your instructor posted your activity sheets yet? Okay. Who's your instructor? Do you know? Okay. There's a link to a neat YouTube video I put in the activity sheets so you can see a dissection. Hopefully go check and see before lab if you can. You can just quick view that. But what you're going to do is you're going to explore the inside of the eyeball, a cow eyeball, which is bigger than your eyeball, and try to identify as many of these parts as possible. So when I look at the eyeball, you're seeing a very small portion of it when I look at you. Most of it is in that nice cranial cavity, the orbit of the eye. And this part that comes out the end is going to carry what? Message to where? What part of the brain? To the occipital lobe of the brain to process vision. So this structure, the eyeball, is designed to get optimal light to fire off optimal receptors so you get optimal information to process in your brain for vision. So that we have, in the eyeball, there's 3 separate layers. There's a really, really thick, fibrous outer layer. And when you dissect your eyeballs today, one of the things I want you to do before you cut into it, give it a little squish. It's like a Super Bowl. It's really tough. This outer layer is called the what? Fibrous layer. And that goes all the way around the outside. Now that fibrous layer has 2 parts. The one that you look at head on, that fibrous layer is clear. Does anybody know what we call that part? I can see through it. So when I look at your eyes and I say you have blue eyes and you have brown eyes, you're looking through this part. It's called the cornea, and it's clear. It's part of the fibrous layer, but it's clear. It's a good thing because light wouldn't be able to pass through it if it wasn't. What's the other part? It's called the sclera, and that's the white of your eye. So the sclera and the cornea are both part of the fibrous tunic. So if I follow this all the way around, I can see it go all the way around. And this part is what? This part is clear. So this is your cornea. Behind that, in the diagram, you see sort of a reddish layer. It's called the what? That's the vascular layer. You can transplant a cornea. Yep. Because sometimes, anybody have astigmatism? You'll test for that in lab today. It's basically a little ripple in your cornea that kind of causes light to deflect in the wrong direction. Some people have such bad deflection that they can't see. So yes, you can transplant a cornea. Think about that when you take your corneas off in lab, because can you imagine having to put it back? Just think about that as you dissect. So it's really a fine surgery, but
They cut around, just around the circle around.
yep, put a brand new one on. I don't know, honestly. I'm not familiar with the surgery, but there's so many different laser techniques that I'm guessing there's probably something that can kind of tack it down. But yeah, cornea, you can do a lens transplant. That happens all the time.
Well, it's kind of the same with cataract.
Yep. And we'll talk about that in a minute. So the cornea, the clear part of the fibrous tunic, below that the vascular layer, which consists of a couple of different parts. So if you look at that reddish portion in the diagram, we see that this is where most of the blood vessels are that are going to help feed and nourish the cells of the eye. We're also going to find, I should bring my pointer in, a little muscular region here, which is an extension that comes up here. One area is called the ciliary body, and this area here, which is pigmented, is called the iris. These are little muscular portions of this layer. Does anybody know what an iris is for? Nope. Well, it doesn't see anything. What sees? Those receptors. I send the information, the brain sees, right? But what does the iris do? Yeah, the pupil is a hole. It's not a thing. I can't take your pupil out. It's just a hole. And it's smaller when the iris is like this, and it's larger when the iris is like this. So when the iris constricts and opens up and dilates your pupil, what comes in more? Light.
So that's why when you walk into a dark room, it has to be
Goes, woo, and you're gonna do some tests in lab that will see what happens to the iris, or the pupil is what you're looking at, with different intensity lights. So now, right now, because I shut the light off, your pupils are probably fairly large, letting in more light so you can visualize what I want you to see. But if I go over there and flick the light on real quick, what's going to happen? I'm going to do it. I'm going to do it. Everybody goes, why? So what happened to your iris? Makes the hole what? Smaller, because you don't need as much of the light from the room to come in and fire off those receptors. It can actually damage those receptors if you get too much light. If you had me in lab, what am I always saying to you? Check your light. If your light's too high, and I say don't burn your retinas out. But if the light is too high, can you visualize the object that's on the slide? Why? It kind of washes out your ability to fine-tune that information that's going to your brain. So you turn down the light or you adjust the condenser to decrease the light. Basically, your microscope is acting like the apparatus of your eyeball. It's acting like your iris. That's why we call it a what? That little knob on the microscope. It's called the iris diaphragm. Yes? So that's what your eyeball is doing with these accessory structures.
So when you go to the eye doctor and they put those drops in your eye, so
Doesn't allow your eye to... Why do you think they do that?
that they can see what you're seeing.
No, so they can see that, the integrity of your eyeball much better. Because if they take that light and shine it in there, what are you automatically going to do? Make a tiny little hole. And it's very difficult for them to get a good look at the whole back of your eyeball. So if they dilate your eyes, messes you up for the rest of the day. But that's why they do it, so they can get a good look at the back of your eye. They can get a good look at the integrity of your retina and your eye inside. So we have the choroid, which is a dark portion of this vascular layer, to draw as much light back there as possible. Light. Does anybody know what light is made of? When I look at white light, what is white light? Is that ringing a bell?
It's a rainbow. It's a red, orange,
Yeah, Yeah, that's what white light is. White light is a combination of different wavelengths of light in these different spectrums. So red, orange, yellow, green, blue, indigo, violet. That's what Roy G. Biv is. Okay? And that is different wavelengths of light that's coming into your eye. So when I see something purple, where is that wavelength of light flashing into my eye? It's in this range, right? Oops. Yes? When I see something red, it's in this range. Okay? So those wavelengths reflect off the object and into your eye. The accessory structures get it to where it needs to go. When I see something white, all of those are reflecting off of the object into your eye. So in the summertime, what kind of shirts do you wear? White. How come? Why do you wear white in the summer? Because it reflects all those wavelengths. They don't absorb into the object. Black. Exactly, the exact opposite. So what you're going to see is what reflects off into your eye. If you reflect a lower or more different combination of wavelengths, it's kind of like crayons. If I put a whole bunch of different crayons together, I'm going to get black or brown, right? So that's why you see the colors you see. And black is going to attract a lot more of those different wavelengths to it and absorb it. And that's what we find in the choroid layer of the vascular layer of the eye, attracting as much of that light that's bouncing off the object as possible so that it can fire off who? Fire off the receptors. And what kind of receptors are these? Photoreceptors, because what's the stimulus? Light. In all of these different wavelengths. The ones that work on our receptors are called what? No, you go ahead and ask your question. When we don't have proper chemistry associated with those photoreceptors. So we're going to find there's different combinations that they'll be fired off in based on the wavelength of light that hits them. When the chemistry is off, we're not going to be able to make those combinations down in the brain area, and that's what color blindness is. So the cones or the color receptors are off chemically. You might have an absence of them, or you might have a chemical deficiency of them. It won't allow you to process that information properly. So, what was I saying before? What was the question I was going to make you answer that I didn't make you answer? Nobody remembers? What was? We were talking photoreceptors. We were talking, I don't remember. You remember when you go back and listen to the, anyways, so we have the, where were they located? I think that's where we were going. So, all of these different photoreceptors, and we have groups of two different groups. We have ones called cones, and they're going to pick up the different colors of the spectrum. And then we have ones called rods, and they're basically for gray tones. I kind of call them our black and white TV receptors. They're located in which layer of the eye? Now we're talking about the third layer, the yellow layer. What's the yellow layer called? Nope. The retina. So we see the fibrous, the vascular, and the retina, or some textbooks sometimes call them the sensory tunic. Your textbook calls it the inner tunic. But this is where the receptors are located. They're embedded in the retinal layer. We also have some pigment on the back of it, again, to draw as much light as possible. Then we have the neural layer where the receptors are actually located. Yeah.
So when like boxes get detachment, it's a whole piece just falls off the.
It pulls off, and then it's going to disrupt the message that gets sent down the optic nerve.
Get like going from here.
Yeah, and it can disrupt. We're going to look at the retina, its cellular makeup in 1 second. But if this portion, say, pulls off, or more importantly, this portion pulls off, there's cells located in this little area. This is where light wants to go. And that's what all your structures are going to do, is direct light to that point. It's called the macula, luteae, and fovea centralis. So if that pulls off, I'm going to disrupt that neuronal signal and disrupt my vision. And we'll talk about what the retina looks like in a minute so you'll understand. All of the message is going to get sent here. What do you think these yellow things are? They're parts of neurons. A whole bunch of them in one place is called a nerve, and this is the optic nerve. How many branches of the optic nerve do you have? Because you have what? 2 eyes. They're going to meet in an area called the optic chiasm, and then they're going to split, and some of the pathways are going to go to the right side, some of the pathways are going to go to the left side of your occipital lobe. And again, this is where vision is going to be processed. So if I look at the cellular makeup of the retina, I'm going to see in 1 second that there's tons of different rods and cones and different patterns. And in that area called the fovea centralis, or macula lutea region, this is where I'm going to find the highest concentration of cones. This is where I have my highest visual acuity. What the heck does that mean? Well, all right, I'll give you an example. I'm going to stare at you. Are you the only thing I can see? Oh, I can see you behind her going, and turning his head around. I'm still looking at you, but I can see him. I can see him all the way over there. I can see her. She just lifted her hand up. But you are in my what? Best focus. The highest what? Visual acuity. So I have all this information bouncing off of you, coming into my eye, but where you're sitting, because of where I've positioned my eyes, the information that's bouncing off of you is hitting where? Right there, at the highest point of visual acuity. I have all my other receptors firing off, but everything's a little bit what? A little bit fuzzy. But you, perfect. Yes? Nope. The axons of these neurons, as they send their message, will then send their message out the optic nerve. So these are basically the axons of some of the cells we're going to talk about in a minute. Sending the message I just got, the information bouncing off of you with respect to light bouncing off of you in different patterns. So you have a yellow shirt on and a grayish coat on with a brown collar. How can I tell that? Yeah, they're firing off in different patterns. Those photoreceptors are bringing that information where? To the brain for processing. You getting it? You with me? Okay. So it's the different patterns, different wavelengths of light, coming in different patterns, firing off different patterns of receptors, with the help of what? What is this? Up there. This is what? This is your iris. So there's a series of muscles that are going to control the amount of light that comes in to fire off the receptors in different patterns. And this is probably where I'm at now when I look at you, because the room's kind of dark, right? Who's kicking in to help create this large opening to gather as much information as possible? The sympathetic division of the what? Of the autonomic nervous system. Do I have to sit here and think about what I have to do with my eye muscles when they're like, oh, it's dark, I must quickly, sympathetic nervous system has to kick in. It happens what? Automatically. So it's part of the autonomic nervous system. When I flick that light on, the parasympathetic division of the autonomic nervous system is going to kick in and cause less light to come in. Again, I have to have a certain pattern and I have to have a certain intensity in order for me to fire off just the right receptors so I can see properly. So sympathetic and parasympathetic divisions of the autonomic nervous system are going to help bring that information in. The iris itself has pigment depending on your genetic makeup. So some of us have blue irises, some of us have brown irises, and then a whole bunch of different shades in between. We'll talk about that when we talk genetics.
Our eyes change colors.
They don't necessarily change colors, but they reflect differently, depending on what? What you're wearing. What you're wearing or the intensity of light you're in. So in the summertime, your eyes tend to look a different color than they do in the winter time.
Well, this morning I woke up and they were really green, and normally they're hazel or brown.
Yep. And that, again, depends on what you're wearing, what intensity light you're in. And the other thing, believe it or not, that can change what you reflect is the chemical makeup that creates that color. And what do you think that might be? What can change your body chemistry? What you eat, nutrition. So sometimes, say in the summer you tend to eat more fruit, let's say. You might have a different intensity of what you reflect eye color-wise than you do in the wintertime. Yeah. And again, remember, it's the intensity of the message you're sending off that people are picking up. Sometimes when you look in the mirror, your eyes look a little lighter to you than they actually do because you're getting information secondhand, right? Your eyes are reflecting their color to the mirror and the mirror is bouncing it back to you. That makes sense? All right, so this is the cellular makeup of the retina. And remember, this is a blow up back here of what the cells look like. So when I look at the retina itself, it's made up of several layers of cells. Now this is the confusing part. Light comes in this way. So we're looking at the back of the eyeball, yes? Light bypasses all of these cells because these aren't what? They're not photoreceptors. They don't get fired off by light. What do they get fired off by? No, not the light, but what these guys are going to give off and send back. Neurotransmitters, very good. So these guys are more chemoreceptors. So the light's going to bypass them and nothing's going to happen until the light is attracted to this back layer and closer to who? The photoreceptors. These purple long things and the little yellow triangle things, those are the photoreceptors. The purple ones are rods, and these are what? Cones. So these are the photoreceptors. So light is going to pass through all of this other garbage, and nothing's going to happen until it hits these guys. These guys are going to be stimulated by the different wavelengths in light in different patterns. And different ones will fire off and send message where? Back in this direction. So these cells, called horizontal cells, and these cells, called amacrine cells, will fire off in different combinations based on message they get from these guys, the bipolar cells, which then will pass message on to the ganglion cells. And the axons of the ganglion cells are all of this yellow stuff. That's going to bring message where? To the brain for processing. So the photoreceptors don't bring the message. They're just the initial stimulus, the initial cell that then passes the message in different patterns. That's why we can see so many different things, so many different patterns, so many different color combinations, because we're firing off different numbers of those photoreceptors and then passing that message in many, many different patterns through these groups of cells to the brain for processing. So you need to understand that light comes in and bypasses these guys, eventually fires off the photoreceptors. Message then gets passed back through the bipolar and ganglion cells, and then the axons of those ganglion cells are what runs down the optic nerve to process sight in the occipital lobes of the brain. Now, there's a little place right around where all of these guys kind of feed in together. Notice there's no photoreceptors there. Did you notice that? What's that called? It's called the blind spot. So right now in my field of view, I have two eyeballs that are bringing in information and helping me see what I see. Right now in my field of view, there's a spot that's blank. There's nothing there. How come I can't see it? Because there's nothing there. Very good. No, there's things there, but I can't see them. Why can't it? Why doesn't that like scream in my field of view? Yeah, your brain compensates for that. You're also going to test in lab to prove to you that you do have a blind spot. Have you ever been driving and you go to change lanes and you kind of look over and there's nobody there, and then you go to change and the guy behind you is beeping his horn at you and swearing at you? Yes? Because that was probably in your what? In your blind spot. So there is a spot in your field of view where you have no stimulus. And it's right where the optic nerve feeds out the back of the eyeball. It's called your blind spot. So you'll test for that in lab and you'll actually see that you do have a blind spot. So understand what that layer consists of. Understand the pattern of light going through the cells, firing off the photoreceptors and coming back. Those axons of ganglion cells are the ones that are then going to bring message via the optic nerve to be processed in the brain. Within that optic nerve, you're also going to have blood supply. So we'll see arteries and veins there as well. And those are going to supply food to the cells, the axons of those cells. When I look inside the eye, and unfortunately we don't have an ophthalmoscope in lab, so we can't do this in lab. I do have some neat pictures though, so ask your instructor to pull out the pictures from the box. Has anybody had this done when they go to the eye doctor, Sue? Did they ever show you your picture? Yeah, they have a retinal scan. Now some of them actually do a separate retinal scan. I know it costs some extra money, unfortunately, over and above your insurance, but I highly recommend you do that, especially if you have any vascular issues or history of vascular issues in your family. You don't want to get a detached retina and not know about it. So these examinations are really important. Diabetes, for example, can cause small little bleeds in your eye that can be picked up by that. Did you have a?
Yeah, I have diabetes, and so when it's high, I feel
Yeah.
my eyes.
Or blood pressure issues. Anybody with high blood pressure or history of high blood pressure in the family, it's a nice little extra thing to do because it can help keep the integrity of your sight. So what they do with this is they have a peek in the back, and that's sometimes why your eye gets dilated, so that the doctor can have a good, thorough view of the back of your eye. You can see that macula. It's kind of a darker region here where the fovea centralis is. And what was that again? Most concentrated area of cones. This is your highest visual acuity. And what's this? The blind spot. Another name for it is the optic disc. And then what are all those lines all over the place? Yeah, that's your blood supply. So when they go in there and have a peek, that's what they're looking at. So when they shine a light in there, how come it's red? It's the blood supply back there. You see a black pupil because there's no light in that room. What happens sometimes when you take pictures? You get that red eye? That's because when you shine that light in, it just happens to bounce right back out the hole, and you pick it up on your camera at just the right angle.
How come it seems like some people are more prone to
Because it's the angle at which that light hits. So certain height people, depending on who's taking your picture, tend to have more red eye than others. Probably, you're probably right. Yeah. So where the angle is that they're picking up that picture. So that's what they look at when they go to the doctors, to check the integrity of all of those sites. Macular degeneration is when those cells start to break down.
So that's where it happens, is
Correct.
concentration.
Right. And what happens to a person undergoing the process of macular degeneration? What happens to their sight? Do you know?
It's really blurry and cloudy, right?
Yeah, but
Dark.
it starts in an area and it moves out. So you'll start to have, all of a sudden you'll have your peripheral view, because all of those guys are okay, but that center starts to go away. So when it's really bad, you'll start to see a black spot and then all the other stuff around it. That will start to become fuzzy in the beginning.
When I get a migraine and I get tunnel vision,
Mm-hmm.
so is it really just, is it like a misfire that's just not allowing this stuff to work?
No, it's just firing off receptors that normally would be fired off by light. So sometimes you get those visual auras where you actually start to see light. Like I just before a migraine, sometimes people will get, will get floaties, or all of a sudden the edges of their vision will start to get twinkly.
that is such, that happened to me at work.
Yeah.
I was dying.
Yeah. Well, it's scary because the first time it happens. It happened to me once. I was in my son's kindergarten class, and all of a sudden it's like, okay.
Like at home, I Googled it, did like rainbow flashy lights on the side of my vision, it came right up.
Yep.
The thing was,
Because that's very, very common, and it has to do with the blood vessels constricting and firing off receptors.
I never
When
got a migraine.
no, I got kind of a little bit of a headache, but not a migraine. Yeah. So it'll happen for different reasons. Another thing people might get is smells.
It almost made me pass out too. It was freaking out.
Right. Well, and then you start losing your perspective of where you're going, and things, which is kind of important. But yeah, so yes, that happens.
It started like this, and then eventually it was a whole.
Mm-hmm. Or around the edges, people get them in different, very distinct patterns. So it's very common to have that happen. All right. So that's my retina. I understand all of the patterns and all of that good stuff, and what the fovea centralis is, and the macula luteae, and rods and cones. Now, another thing that's very important is, remember, getting this light to where it needs to go. And when we talk about the external structures that are going to help with that, not only do we have physical structures, but we also have fluid and fluid compartments under different pressures. So that when they talk about the chambers of the eye, there's two chambers. We have one in the front, and what do you think we call that? Anterior, and we have one in the back, posterior, that are filled with different types of fluid in different consistencies. Behind the cornea, before the iris, we have the anterior chamber. And in the anterior chamber, we have a fluid that is very much the consistency very similar to water. That fluid is called the aqueous humor. And that fluid is constantly being produced by these groups of cells in the ciliary body and floating and rotating to help keep a certain pressure within the anterior chamber to help that light get to where it needs to go. Do you ever go to the eye doctors and they do that extremely annoying little puff in your eye, which you know it's coming, you know it's coming, and you always blink no matter what. What are they doing besides annoying the living daylights out of you? They're testing the pressure in the anterior chamber. When pressure builds up to too much, it's going to start damaging a lot of the structures, and it's going to start inhibiting your vision.
Certain eye medicines.
Glaucoma.
Certain eye medicines that you take for different things can mess with your pressure too.
Correct. So I'm constantly making it, and it's getting reabsorbed. This area here called the scleral venous sinus is where we reabsorb that liquid that's being produced. Unfortunately, I don't know what happened to my diagrams. They got all messed up. But this is on page 552, figure 15.8, and it shows you the circulation, production and circulation of aqueous humor. We don't want it to build. If there's a blockage of some kind that causes too much pressure, it can start damaging structures there. And again, there's a little, I thought they did it. Homeostatic imbalances on page 553, they talk about something called glaucoma. And again, that's too much pressure building up in that chamber that can cause problems and eventual blindness if let go too long. Yeah, whatever the eye. I'm not familiar with what the measurement is. Is it? And some kind of blockage of that flow of fluid. Just before the back chamber, there's another little structure. It's held in by some of these little ciliary bodies and these little suspensory ligaments, and it is your lens. And what's a lens for? Again, it's going to help get the light to where it needs to go. Not to be gross or anything, but and I know some of you people have done this, so do not judge me. The little hand lens, burning ants out in the backyard. Yeah. Yeah, you probably have, right? No? Yeah. Why does that happen? Because the lens is helping to do what? Concentrate the rays of the sun to a place where they can be very, very hot. Actually, I first learned to do this at camp. We actually had a piece of wood, and they made us, we use it to, you like draw a picture on it. So we tried to, yeah. But then we got bored with that, and it's like, ooh, ants. So the lens is going to help, again, concentrate that light to go where? Where do we want to get it to go? To the macula luteae, fovea centralis region. So that all of these are going to help direct that light there. So we'll pick up with the posterior chamber on Thursday.